Assalamualaikum and hi everyone. So today we are going to present a cytology and cytogenetic practical presentations, and my group members are me myself Azman bin Abdul Karim, Siti Nobaya, Najwa Najiha, Nur Shalin Zuhaila, Fazrina, Ong Sun Liang, Nur Fahana and Umu Asma.
Next, for brain cells, the technique we used is impression smear. Then, the slide is stained with PEP stain. Based on the microscopic investigation, there are neurons, astrocytes, and oligodendrocytes. Neurons, as shown in the black circle, have large and round nucleus. Meanwhile, astrocytes, as shown in the blue circle, tend to be larger with round to oval shape and have less dense chromatin. Oligodendrocytes, as shown in the green circle, on the other hand, are smaller than astrocytes. However, both oligodendrocytes and astrocytes are basophilic. These are a few examples of neuron, astrocytes, and oligodendrocytes that we have found on the internet for future reference. Moving on to lung cells. The technique that we have used to obtain lung cells is impression smear. Then, we have stained the slide with PEP stain. Based on the microscopic investigation, we have found both mature squamous cells and immature squamous cells. Mature squamous cells, as shown in the blue circle, is pink in color. Meanwhile, the immature squamous cells, as shown in the black circle, appear purplish.
Types of organ used is eyeball, which is cornea pipe. Method used is impression smear with May groundwork GMSA MGG staining. For normal appearance of eyeball is dense connective tissue membrane covered with an external non-keratinized stratified epithelium. Cornea is not vascularized and presence of goblet cells on the corneal surface. For picture in lab is using MGG staining with magnification times 40. Arrows show presence of goblet cells on the corneal surface. Also the same with actual picture. Using periodic acid shift staining, arrow also show impression cytology of the conjunctivalized corneal surface showing reddish pink goblet cells.
Moving on to buccal cells. Buccal cells are collected by scraping the cheek with a cotton swab using a technique named buccal smear. The swab is wiped firmly across the buccal area and then rolled against a glass slide so the cell adhere to the slide. PAP stain was used to observe the cell. In this preparation, squamous epithelial cells from the oral mucosal stratified squamous non-keratinizing epithelium is observed. The normal cell have more cytoplasmic than nuclear area. The nuclei of normal cell are usually round or oval shaped. Also, the epithelial cells will be the largest cell in this smear. This is the comparison between actual picture with picture taken in the lab. The observation is considered as succeed because the buccal cell taken in the lab has similar cytology as actual picture of buccal cells.
Next is the self-hearing collection. For this part, we have collected clear cache urine sample. We were using voided urine and then the urine was stained using PEP staining technique before we can observe under the microscope. The appearance of normal urine must be containing urothelial cells. Other than that, in women, the squamous cell from the vagina may also be present. Another important thing for normal urine is that there is no presence of malignant cells identified. So when observed under the microscope, we are expected to see some urothelial cell covering the surface of urothelium, and normal columnar urothelial cell may also be present. In the actual lab, the cell observed were not really clear, but we can still see the clustered urothelial cell and presence of few squamous cells. Move on to the next part. We will also taking sample from the thymus. The method used for thymus was impression smear and then stain using pap staining technique. The normal appearance of thymus may include the presence of epithelial cells, densely packed immature lymphocytes, and we may observe the presence of lymphoblasts. So when observed under the microscope, we are expected to see centrally located loose clusters of epithelial cells which are easy to recognize. And then also the rounded to slightly oval nuclei and absence of malignant features. In the actual lab, when observed under magnification of 40 times, we can observe lymphocytes in a dissociated pattern. And then there are also larger cells with identifiable cytoplasm that may represent the epithelial cells.
Kidney and heart cytology were obtained by using an impression smear and stained with pap stain. The picture shows cellular morphology of kidney cells. The picture on the left demonstrates the glomerulus appeared as dense cellular spherical structure with scalloped edges. The glomerulus also shows like atypia, and they appeared denser at the center. The tubular cells can also be seen in kidney cytology, where features of proximal tubular cells are distinct to distal tubular cells. The nucleus in the proximal tubular cells is round and bland with the presence of prominent nucleolus. However, the nucleus in distal tubular cells is smaller compared to the nucleus in proximal tubular cells with inconspicuous nucleolus. The cytoplasm in proximal tubular cells appeared granular and abundant, whereas the cytoplasm in distal tubular cells is lesser compared to proximal tubular cells, and they are clear. The membrane also shows distinct features between the proximal tubular cells and distal tubular cells, where the cells have poorly demarcated membrane in proximal tubular cells, while distal tubular cells have well-defined cellular membrane. In the impression smear of kidney cytology, there are no glomerulus obtained and only the tubular cells are observed. The cells show abundant granular cytoplasm with poor defined membrane and round nucleus. Next, the picture demonstrates the cell morphology of cardiomyocytes, or the myocardial cells with large nucleus and slightly scant cytoplasm, but with well-defined membrane. The next slide shows the myocardial cells obtained by impression smear with well-defined cell border and clear cytoplasm that takes up blue-green color, while the nuclei are stained blue and appeared large.
So, next we move to the cytology of the liver. Any study of the cellular composition of fine needle aspirates from the liver requires an understanding of the liver structure. In brief, the liver consists of small hexagonal lobules, each with a central vein. The liver cells, hepatocytes, are arranged in thin sheets which radiate from the central vein to the periphery of the lobule and are separated by sinusoids lined with endothelial cells and phagocytic Kupffer cells. So, here's the result we obtained in our practical session and what it should be looks like in the normal condition. This is the fine needle aspirate FNA of the liver and stained with MGG stain. The results were then observed under light microscope with 40 times magnification. In the figure A, we can observe that the central parts of smears prepared from the normal liver aspirate contain mostly blood. Besides, from both of the slides, we can observe that the cytoplasm stain blue with MGG stain and its structure is irregular reticular. Dark blue pigments granules can then seen in many cells, as shown with the arrowhead. Furthermore, the cytoplasmic border between the adjacent hepatocytes are not distinct as shown of the arrow. Next, here's the fine needle aspirate FNA of the liver and stained with pap stain. The results were then observed under light microscope with 40 times magnification. In here, we can observe that the aspirates of the normal liver consist predominantly of hepatocytes. The hepatocytes are present as single cells or monolayer small cells groups and sheets. The cells are round, polygonal, having a well-defined cell border and granular dense cytoplasm as shown by the arrow. Besides, in figure B, a small cluster of tiny bile ductal cells in a honeycomb pattern is present as shown by the arrowhead. Bile duct cells are sparse in normal cells and normal liver aspirates. They usually have less cytoplasm and contain round to oval nuclei with indistinct nucleoli.
Vagina flushing. It is located at vagina. We use the method of impression smear. Papa-Nicolau staining and May-Grunwald Gemsa staining is the type of stain that we use. The cell morphology are the cells are in densely packed nucleated cells and superficial nucleated epithelial cells with cytoplasms. For Papa-Nicolau staining, the picture in the lab with a magnification of 10 times show the presence of the nucleated epithelial cells with pink cytoplasm. For the actual picture, it is show the presence of densely packed nucleated cells with pink cytoplasm. For May-Grunwald Gemsa staining, the picture in the lab with magnification of 40 times show the presence of the nucleated epithelial cells. And the actual picture show a smear with densely packed nucleated epithelial cells.
So that's all from us. Thank you.